This video is going to demonstrate the gram stain. So the gram stain is a common cytologic and actually microbiology stain that we use to differentiate gram-positive and gram-negative bacteria. This technique was actually developed by a scientist named Christian Graham, who in 1884 found that different types of bacteria actually had different cell wall characteristics. If their cell wall contained peptidoglycan, we found that these cells or bacteria would actually stain gram-positive or retain that very vibrant purple color, versus if they were considered to be gram-negative or lacked peptidoglycan or did not have an abundance of peptidoglycan in their cell wall, they would retain the counter stain, safranin, and result in a brilliant red color. So to perform a gram stain, you're going to need several supplies. So first would be your actual specimen that you're wanting to gram stain on a glass slide. And then your 4 components of the gram stain. First, your crystal violet. Second, your iodine. Third, your alcohol decolorizer. And lastly, your safranin, which is your counter stain. You'll also need blotting paper, as well as gently running tap water. The first step of preparing a gram stain is to apply your sample to a clean slide and then heat fix the base of the slide with a flame. The slide should feel warm to the touch, but not hot. Second, you're going to flood the slide with the crystal violet solution and allow it to sit for 60 seconds or 1 minute. Once it's completed staining, you're going to rinse the excess stain off gently and briefly with tap water. The tap water should just run clear. Then you'll blot the excess water off of the sample. Next, using the iodine, you will once again flood the sample for 60 seconds with the iodine solution. The iodine combines with the crystal violet and fixes it to the cell wall. After 60 seconds of time has elapsed, you will once again rinse the excess iodine off briefly with the tap water and blot it onto your paper towel. The third step can be the most challenging step, as if you over-fix it with the alcohol, you can lose your color. You're going to place your sample into the alcohol fixative for 10 seconds and then rinse gently with tap water. If you have a smear that is excessively thick, it may require longer for decolorization. It requires some experience to know how long to decolorize your slide for. Note the decoloration will only occur on the gram-negative bacteria, while the gram-positive bacteria will retain their crystal violet or purple staining components. Finally, in order to visualize the gram-negative bacteria, we'll apply a counter stain called safranin and allow it to sit for 30 seconds. Once 30 seconds of time has elapsed, you'll once again rinse off the excess stain briefly with tap water. You have now completed your gram stain after you have blotted off the sides of the slide for excess water and allowed to air dry. You can also blot with biblious paper. However, you must be careful not to remove the sample from the glass slide when you use this mechanism. Also, do not rub the sample off of the slide using paper towels or the biblious paper. Lastly, you'll now view your slide under the microscope at both 10x objective and then going down to 100x or your oil objective, looking for your purple or gram-positive bacteria or your pink or red safranin dyed gram-negative bacteria. It is important to remember that gram-positive bacteria whose cell wall has been damaged or degrading, most commonly from antimicrobial or antibiotic administration, or from being attacked by phagocytes, will likely appear gram-negative because they have now had their structural integrity of their cell wall destroyed.